In this video, I want to talk a little bit about the anatomy of central synapses, so synapses between two neurons found in the brain or in the spinal cord. If you watched my previous video on the neuromuscular junction, you know that that was a very specialized synapse, and there were a number of things that made it special: a single nerve cell, a single target cell, and another thing was the anatomy. We can see that here. So here's a muscle fiber, a single muscle cell, and here is the end plate where the motor neuron is innervating it. And although when you look at this section, you can see that there are several other end plates around, they're really pretty far apart, meaning that the signal that's released from this nerve cell really can only be seen by the target cell. The situation in the brain is pretty different because it's very crowded. We can see this in this Golgi-stained section of a mouse cerebral cortex. I've added some circles here to try and show you the size of the dendritic fields, and I hope you can see that their arborizations are overlapping. Remember that in Golgi staining, we only stain about 1% of the cells, so there are even more cells there and even more dendrites that might all be overlapping one another. You can get a pretty good sense for that if you look at a Nissl stained section. So here again is cortex, and here each one of those blue dots is representing the cell body. So that raises a question then, okay, it looks really crowded. How many neurons are present in the brain, and how many synapses are each neuron getting? Well, this has been addressed by a number of researchers, including by De Felipe and his colleagues, and I'll show you some of that data. So what they did in a study was to go and take sections of tissue. In this case, they were looking at a 50 micron by a 50 micron column of tissue, so 1/20th of a centimeter by 1/20th of a centimeter, and the cortex in the place where they were looking was about 2.6 millimeters tall. So they took this tissue and they counted the number of cells and counted the number of synapses. And what they found was that there were about 158 neurons and a whopping 5 million synapses. And that works out to be about 30,000 synapses per neuron, so much, much more complicated than at the neuromuscular junction. We can see this if we look at electron micrographs. So this is an image that I took from BU's Atlas of the Brain. It's from a monkey. And what the authors of this atlas did is they color-coded the different structures in the image so that green represents axon terminals, blue represents dendrites, gray are dendritic spines, yellow are astrocytes, that's a type of glial cell, and uncolored things are then axon profiles. And if we take a little chunk of this and we blow it up, so you can look at this in even more detail, you can begin to see the synapses. Here's a dendritic spine, and on one side, here's an axon terminal, and it's making a synapse onto that dendrite. Here's another axon terminal, making a synapse onto that dendrite, and the structure is surrounded by these glial cells, by the astrocytes. Now, it looks crowded, and if you look, you can see there are a whole bunch of other dendrites present, and they might be dendrites from the same neuron, they might be dendrites from a different neuron. There are a whole bunch of different axon terminals making synapses, and there are a whole bunch of glial cells. Electron microscopy studies have also revealed something else about synapses, and that's that there are two different morphological types of synapses. And to illustrate this, I have to take away my the marks that I put around the cell membrane. If you look at this synapse right here, it's a symmetric synapse, and the way that we tell that, and the way that it gets its name, is by looking at the pre- and the postsynaptic densities. And what you can see if you look at the lines in that circle is that they're about the same intensity. The presynaptic cells got lots of vesicles. I haven't colored them all, but the ones that I have colored show you that they appear a little bit flattened. And at least in tissue that's been prepared for electron microscopy, when you see flattened vesicles like that, it tends to correspond to inhibitory synapses. And what I mean by that is that if that synapse is active, then the target cell is less likely to reach its threshold. The other type of synapse are called asymmetric synapses. And here the postsynaptic density is more prominent than the active zone in the presynaptic. You can see that I've colored it in a little bit, and I hope you can see that the blue side is fuzzier than on the green side. Again, we'll have vesicles in the presynaptic cell, but here they look more round. And synapses with this kind of morphological set of features tend to be excitatory, meaning that the target cell is more likely to reach action potential threshold if that synapse is active. Now, these two types of synapses can be seen other places. So, for example, if you look at the synapse down on the bottom right, you'll see the fuzziness and the round vesicles. It should make you think that this is an excitatory synapse, an asymmetric one. Well, let me go back then to this study that De Filippi did, and we can think about, well, how many of the synapses that a neuron gets are excitatory versus inhibitory? And based on these morphological characteristics, it looks like about 80 to 90% of synapses are excitatory in the cortex. Different parts of the brain are different. And about 10 to 20 are symmetric or inhibitory. It isn't the same in all layers of the cortex. So up in layer 1, that's where you have the highest number of inhibitory synapses, whereas in the heart of the cortex, it's about 90% excitatory, 10% inhibitory. So this can vary as well. And it can vary also between different animals. Okay, I'm going to turn now to a cartoon just to introduce a little bit more terminology. So my cartoon here is of a pyramidal neuron, common cell type in the brain, and I've drawn it with spines. These neurons can get input, and in fact the spines are the site where there are synapses. And often what happens is an axon will come and it will terminate on the spine. We call these axodendritic synapses. There are other axodendritic synapses where a single fiber will pass through a row of cells, and as it does that, there'll be swellings where neurotransmitter can be released. These are called en passant synapses. Well, whether we're looking at the en passant synapses or the ones where the axon terminates onto a spine, these, in mammals at least, tend to be excitatory. They're not always excitatory. I showed you an example in electron microscopy where there was a synapse on the spine that was inhibitory, but they tend to be excitatory. In addition to these kinds of synapses, there are other morphological types. For example, sometimes axons come and they terminate onto the cell body. We call these axosomatic synapses, and again, in mammals, these do tend to be inhibitory. And finally, we'll have types of synapses that are called axo-axonic synapses, and these come in a number of different varieties. Towards the top of the figure, I have one where the axon is terminating onto the axon terminal, and then lower towards the bottom of the left side of the figure, you can see one where the axon's terminating onto the axon initial segment. Whether you have the axo-axonic synapse onto the terminal or onto the spike initiation zone, these tend to be modulatory, meaning that they can affect the release of transmitter and the propagation of action potentials. Well, we need to know a lot more about how nerve cells integrate information at both excitatory and inhibitory synapses in order to understand how they're going to be able to add up all the inputs that they get and then make decisions about whether or not to fire action potentials. And that's the focus of my next video.